let's start here. Could anybody just remind me, give me a brief overview of what phagocytosis is again? What is it really? Simple description of phagocytosis. It's a process by which anyone.
Um, this is by which white blood cells known as phagocytes engulf and destroy pathogens.
Right, so engulf and destroy pathogens. Lovely. Right, so it's usually what we call so they're gonna, well, it's generally just engulfing, right? They're gonna use their pseudopoda and their different phagosomes and stuff like that with their lysine enzymes to actually break up the pathogens, right? So we're gonna have firstly chemotaxis and recognition, right? So chemotaxis is going to basically be the movement, right? Taxis is movement, chemo is chemical. So what we're gonna have, right, is that when there's some type of injury, right, or any type of immune response happening, there are gonna be signals sent out within the blood, within the lymph as well, right? That causes phagocytes to move towards the site of infection or the site of injury. So we're gonna have chemotaxis, right? So they're gonna be moving towards the area, and that's one of the use of complements. We're gonna talk about it, right? So complements actually, when they're released, when T cells release complements and stuff like that, right? It actually calls phagocytes towards the area, right? So the phagocytes, right, will now go and recognize these pathogens, right, through their specific receptors, and then actually start the entire process of phagocytosis, right? So we're gonna have adherence, right? So what we're gonna do, right? Well, what the phagocyte is going to do is it's gonna recognize the target first, right? That's what cellular glycoproteins are for. They're gonna recognize, right, the pathogen first, right, by binding to its receptor, right, and then that's generally what it's gonna do first, and then we're gonna look at what happens after. So it binds to the receptor to figure out what it is, right? So we're looking at the fact that these different receptors that exist on the cells allow for cell communication. One of the major things we looked at in Unit 1 as well, right? So we're going to bind to these receptors, and then after figuring out that this is a pathogen, right, it's going to actually have engulfment occurring. So its pseudopodia, right, would actually um, be projected around the pathogen, right? So we're gonna have pseudopodia, right, formation. The phagocyte extends its cytoplasm around the target particle, forming an arm-like structure called pseudopodia, right? So the pseudopodia eventually encircle the entire pathogen, right, completely engulfing it, right? And the membrane-bound vesicle, right, known as a phagosome. So once it engulfs it through endocytosis, right, it's going to create what we call a phagosome. Alright, so this is phagosome. Don't call it a food vacuole. I just use this diagram because it makes more sense, right? It's a phagosome, right? The phagosome formation there, so we have that entire thing there. So the phagosome is going to be formed, right? And we're gonna have the maturation of the phagosome, right? And the process that acquires various proteins, right? That becomes increasingly acidic. We're gonna talk about what happens within the phagosome, right? Within the phagolysosome, no, we're gonna have the phagolysosome formation, in which we're gonna have generally the phagosome, where it is basically this vacuole in which the pathogen is, right? And then the lysosome will now fuse to the phagosome. Could you guys remind me the use of a lysosome again? What does a lysosome do in the cell?
Produce digestive enzymes.
Okay, lovely. Produce digestive enzymes, right? So that when the lysosome fuses to a phagosome, it becomes a phagolysosome, right? So the lysosomal enzymes such as proteases and lipases and nucleases will now enter into the phagolysosome and degrade all the material of the foreign particle. So it's seen here. So the foreign particle is there, and then it's going to be broken up, right? The complete digestion and destruction of the entire thing, right? So all different types of species, right, are gonna be destroyed. So the digestion enzymes breaking down the pathogenic components, right? So they're gonna be basically using reactive oxygen species and those type of things to destroy the general proteins, right, within the pathogens themselves, right, completely destroy them. They're present within phagocytes, which are especially neutrophils and macrophages, as we'll see, right, and they actually produce these different reactive species that will now destroy the pathogens, right. And the phagolysosome actually creates an extremely acidic environment as well that helps to kill the pathogen, right? We're gonna have exocytosis, or waste removal is gonna be taking place, right? As the phagocyte expels all residual, all waste products, right? The residual bodies will fuse to the cytoplasm, right? So not the cytoplasm, right? To the plasma membrane rather, right? It will fuse to the plasma membrane, right? Releasing their contents outside of the cell. So we're gonna have them now moving all the products moving outside, right? The different type of phagocytes we looked at neutrophils before, right? With macrophages now, these are basically derived from monocytes, right, within the tissue, and they actually result in it actually produces the response of inflammation, etc. And we also have dendritic cells, right, that are in peripheral tissues, right, that are used as well for adaptive immunity. So generally, what's gonna happen? Microbe is gonna be there, right? The pseudopods are gonna be engulfing it, right? We're gonna have a phagosome being produced. The lysosome binds to the phagosome, creating the phagolysosome. Then it breaks down on the material, and then all the indigestible parts will just be ejected through exocytosis. All right. So what we're gonna have is antigen presentation. So what we're gonna have is what we call an MHC class 1, right? And once we actually have the creation of the entire antigen binding to the complex, we're gonna have an MHC class 2, the major histocompatibility complex, right? So this is what a major histocompatibility complex is, right? So what they're gonna do is take all of this material, right? Usually the material is gonna be presented onto its cell membrane. What the bacteria will do now, or what the phagocyte would do now, is take an antigen, place it onto this complex, usually the MHC class 2 complex, right? And present it onto its cell membrane. So look at that presentation. This is antigen presentation, right? So for example, this is a macrophage, and macrophages are usually the larger ones who would do this, right? So the antigens that were digested from the bacterium are now presented on the major histocompatibility complex 2, right, on the cell surface, right? So we have that there. Sure, that is generally what we're looking at for the phagocytosis component, right? Just breaking them up into different components so you understand each and every part, or we're just reviewing each and every part, to be very honest, right?